Now, this is going to be less scientific, a little bit more philosophical, a little bit more, let's say, clinical. I'm a clinician, I'm a neuroradiologist. I'm confronted with daily problems of babies, families, mothers, fathers. And I would like to start off, maybe here. No, okay. I think we have to be aware that, and that came up during different points this morning, that development is a history of space and time. We are looking at the brain, but this is a cross-sectional time point during development, and we are always seeing processes at different levels happening, and MRI is just giving us one glimpse of it. So our own brain as a researcher is somehow biased. We like to see static things. We want to make things static, but actually everything flows and everything is fluent. Even science is a process, and it's permanent interaction between reality. The questions are changing, and so actually everything flows. And also neuroradiology is flowing. As when I started, there was no neuroradiology for fetuses. So fetal neuroradiology did not exist. We had to develop that, and we had to understand what we're seeing. And we are actually confronted daily with the question, is this going to be a normal baby? Now, this is what we know from international consortia, huge data. You're probably familiar with this lifespan research study on brain development. You're looking at different compartments of the brain throughout the lifespan, and also, and that is based on giant amounts of data here, over 120,000 MRI scans grouped together. They also looked at prenatal life, and they realized, well, we could go with that earlier, and also included prenatal data. However, these were 128 subjects in that cohort. So, well, we can look at that stage, and we can look at, for instance, a fetus at 20 gestational weeks. And that's usually the first thing you're seeing. That's an ultrasound. And I have to say here, as MR specialist and experts and neuroradiologists, I love MR. And I love always to kind of be challenged by ultrasound people and say, yeah, we are better, we see more, but I don't know, you know. They see a lot. Look at the image quality there. They see things differently, but actually ultrasound improved a lot as well. Now, this is the similar baby on the same day imaged on 1.5 Tesla clinical MRI. So this is not the brilliant 3 Tesla or very specialized image quality. That's the daily routine image quality we get of the frontal lobes of such a 20 weeks gestational week baby. As you see with MRI, these layers within the brain are much better seen. And so one of the first initial things which came up is to question ourselves, what is this bright line under the cortical dark plate on T2? What are we seeing here? And here, I think this is a very crucial element. Whatever we are doing, we also have to ask ourselves, even if we are synthesizing images, is there a biological plausibility to what we are doing? And I highly recommend the book by Peter Robin Hiesinger, who was with us in Vienna 2 weeks ago on human brain development. I really got a lot of insights through that. So this is the in vivo image of T2. And somehow we need to understand what's behind it. What is the structural correlate? What is maybe the RNA expression in different zones? And somehow we have to match that to an anatomical, let's say, label. And that's exactly what we are going to hear today. And that is exactly the work of Lucas Arcamon. And that's exactly why I believe this is so important, because you need somehow a space to bring together the in utero, the post-mortem, and the histologic data in order to understand all the segmentations we're doing. For instance, here you see nicely that there is a T2 bright zone just underneath the cortex, also on the data nicely shown by Lucas on the T2 relaxometry here, and that well corresponds just visually, for me as a neuroradiologist at least, to the in vivo data. So this is the subplate. What we are seeing here, it's extracellular matrix rich. There are a lot of water molecules around, so it's bright. Wherever there are water molecules, you can also suppress that. I know you as MR scientists don't like to do like FLAIR. It's not quantitative. It's not really fancy. We love it. We love it because we can see the layers much better. So if you suppress the water signal from the T2 weighted image on an EPI FLAIR, or you also can do a compressed tense FLAIR, you get this layering very nicely, and you see this water-rich subplate suppressed by that signal. What is the benefit of doing that? Well, you see these layers now in this brain quite nicely, and I give you a clinical example. That's 23 plus 5. It's not really hard to see the pathology. There is a region here in the brain, somewhere around here, where the cortex is sort of irregular. But with FLAIR, you suddenly understand how much of the brain is affected. So if you look at the contralateral side here, you see the subplate is very nicely continuous. So it's a dark line underneath the cortex. If you go to the other side, it's suddenly interrupted there, meaning that there is a migrational disorder which gives us more security, more certainty to say this is a cortical malformation, and this has a very high chance to generate epilepsy, and therapy-resistant epilepsy, as you all know, is a huge problem. This is the post-mortem specimen which we have in this case. Now, of course, you can also do diffusion. You see at 1.5 Tesla, diffusion is noisy. There are challenges, but at least you see that at the diffusion sensitized sequence, the subplate also has a very dark signal. It's suppressed, and there is a high diffusivity there. This is another structure around the ventricles which seems to have a little bit higher diffusion restriction. And again, Lucas, with his work using NODI, kind of confirming also the high extracellular water content of that region right there, well matching to the in utero even 1.5 Tesla data. I'm just stressing here how important it is to kind of get this groundwork done for us as clinicians to understand what we are seeing. Now, well, if you can do diffusion, you can do tractography. You heard about that. So I'm a clinician. We are using very plump, simple tractography. It's really horrible for you if you consider our post-processing. But as I said, one major thing is biological plausibility. And it seems to be, to some extent, biologically plausible. This is a case with callosal agenesis, where you see the blue bundles are the bundles of Probst, in green corticospinal tracts, and also, of course, others in that. But what is interesting is if you look at the tracts on this side here, they are stopping in that region. They couldn't be followed there. And together, this is a very old case, as you can imagine, with Andras during his time when he was in Vienna. He implemented the technology how to visualize trajectories on histological slides. Andres could talk on that in more detail, but what is really interesting is that exactly there where the fibers were stopping, we could see that those corticospinal tracts were totally disrupted, and you could see that they were not discontinuous here. So there is a biological reality to what we are seeing. Further, we know also that our results are quite well consistent with postnatal tractography results. The in utero tractography, of course, is not so nice and not so detailed, but for us as clinicians, in this case, it's enough to say corpus callosum is present, no Probst bundles. The cysts in between the hemispheres are pushing against the corpus callosum. It's not a primary developmental issue here. It's just an arachnoid cyst. And that has huge implications for this baby and later on boy. That's Matteo. That's the guy you just saw on tractography. He's 6 years old, totally normal. This gives more safety to the patients, more certainty to those patients to say, well, this is an arachnoid cyst. There is no connectivity abnormality here. Now, we are now looked into the subplate. We looked also in this periventricular zone here. And the next question is what is happening here in detail. So if you look at the so-called proliferative zones of the brain around the ventricle, it might not be enough to look down to 18 gestational weeks. You need to go further. They say there is no MRI before 16 weeks. I would like to challenge that. I think we can do MRI before. It's currently also regarded to be safe. These are 1.5 Tesla old images at 14 gestational weeks, not very impressive. They basically tell you there is a brain. They give you an estimation of the ventricles. However, you can now use at 3 Tesla and also AI-supported case-based readout technologies. You can get this basic image contrast at 15 plus 1 gestational weeks in a normal case. And now if you do a little bit AI fancy reconstruction, you really get more out of this image. If you look here at the subplate region and the reconstruction, you certainly starting to see that there is a layering. And we are talking about the subplate formation phase, which is happening at 15 gestational weeks. So I think before 18 weeks, it's not only the domain of ultrasound, we can go earlier, and I think this will be a challenge for the future. Why? Because these ventricular and subventricular zones are highly interesting. They are human-specific. They basically are the largest zones within all species. Nobody on Earth has such a thick ventricular and subventricular zone as humans. You might wonder what we are doing with that, but in most of the times, at least here when we are doing research, I think we are using it appropriately. This is the in vivo image at 1.5 Tesla. This would be this periventricular zone. And now if you look at the flare sequence, it's also interesting. This zone is brighter than the cortical plate. And probably that has something to do with cell density. There are more cells, there is more signal. If you compare to the cortex is a little bit darker. And interestingly, if you observe that throughout pregnancy, it switches. So the cortex will get brighter on flare and the periventricular zone gets less bright. Now, coming back to Lucas' work, he nicely showed also this zone here around the ventricles, which are the ganglionic eminences. Ganglionic eminences can be a fascinating structure. You have heard about transscience today already. We are looking more at a sculpture than a painting. So a lot of things happening in our brain is just removing structures, transient structures, and the ganglionic eminence is such a structure which will be moved throughout development. I'm not going into these things, as Maria already nicely presented her own work on important work on that, which is the groundwork now for us also to be able to clinically segment these structures. We have segmented, for instance, the ganglionic eminences here, and you nicely can visualize them located in this C-shaped pattern. Now, many other researchers have attempted that. For us, it was rather unusual to see the ganglionic eminence always grow. As we know, we don't have any anymore in the adults, so at some point they have to disappear. So in our detailed segmentations here, it appeared that the ganglionic eminence were decreasing in size despite of overall brain development. Now, again, the question, is this plausible? How can we use that clinically? Well, the biological plausibility we are trying to elaborate, and maybe that could be an area of close collaboration. Roxanne Licandro in the team of Georg Langs, who is closely working our computational image research group, is trying to bring together ex vivo post-mortem in situ data with our in vivo data. So if I think about Luca's algorithm bringing together histo and post-mortem, that could be a nice line of research, bring together in vivo post-mortem histo. I just want to show you how this data, this biologically, hopefully plausible data, is now translated to an actual case in clinical, in our clinical life. So you're looking at a pregnancy at 22 gestational weeks, mild ventriculomegaly, meaning like ventricles 12 millimeters of size. Question, is the child going to be normal? And that's what happens in every obstetric ward, obstetric outpatient clinic. So a family comes there, pregnant woman gets scanned. They say, well, there's mild ventriculomegaly. Don't be upset. Everything will be okay. In 90% of cases, everything will be okay. But there could be an issue in 10%. Now imagine, what is the mother hearing? They're going crazy. They are not hearing, well, in 90%, everything is going to be okay. They're hearing, my child has a problem, catastrophe. So one way to approach it is to, for instance, do fetal MR in that situation. This is not the routine recommendation, I have to say. This is still disputed in medical reality. We are having discussions among obstetricians, radiologists, different people from imaging sites. Is it really valuable to do fetal MR? In my view, yes, but it's not given. Ventriculomegaly is a tip of the iceberg problem. You only see the size change of the ventricle, but the other stuff around it, that's where the true scientific value is, as you know. That is what you get with good ultrasound. You can see the slight ventricular dilatation, but also you can see these little cysts lateral to the ventricles, and those cysts are sitting in a certain region. Now, ultrasound specialists would say, I can clearly see the ganglionic eminences here. I'm not so sure. I would say you can't. But therefore, do an MRI, and then you really can see it. So that's what we did. We did the fetal MRI. You see this hypo-intense structure sitting next to the ventricle, and you can see that these cysts are sitting within this ganglionic eminence. What you now can also do, due to Maria's work, you can do slice-to-volume reconstructions, and you can also kind of reconstruct those ganglionic eminences in 3D. And this is the comparison between below the normal case and upside the case, what I'm talking about. And you see the ganglionic eminence up, the upper one, is really thicker, just visually thicker. It also includes cysts, but it's also visually thicker. Now, if you do segmentation, here we are. We are kind of we have double as much ganglionic eminence as expected for that gestational age. No doubt that there is something profoundly wrong going on in this case. While we are doing fetal MR, at the same time, whole exome sequencing is done, usually in most of those cases. Takes 3 weeks. With fetal MR, we get more information. We are a little bit ahead of time when the whole exome is coming in. Now, the whole exome came in, and it was an MTND6 gene mutation. This is a mitochondrial disorder, a generally severe condition. But if you look at the clinical spectrum, we are still not there to say all of these children have a huge, massive problem. You see, not all, it's not 100%. It's like many of them, 50% almost, have seizures and so on and so on. But that's much more information you can give to a mother than just saying, you know, in 10% could be a problem. So that's where we want to go. And we, of course, also want to build a base for everything which comes after. And that is, for instance, gene editing. This is a new project where they are doing particularly gene editing in mitochondrial disorder. In order to do that, you have to catch those cases as early as possible. And that's what clinical MRI is able to do. And I think this is really now appreciated by the medical literature. The really next frontier is in utero. One thing which is a little bit getting under-recognized is that most of us think of brain malformations to be genetic. So there is a gene, has to be a gene, which is abnormal. What we under-recognize is that many of the fetal disorders are acquired. So having a normal genome is not end of the story. There are a lot of acquired conditions which can still make our life very, very complicated. Now, one of those are all the toxic things which are going into our brain. One is fetal alcohol. We did a study on that and looked at alcohol-exposed babies and non-exposed babies, and also using all the tools presented here before, and segmented the periventricular zone in those. And you can see fetal alcohol syndrome or fetal alcohol spectrum disorder, they often have a microcephaly. All these cases which we were looking at did not have microcephaly, but still the periventricular zone, there where all the neurons are born, was too thin, was too little. What was surprising to us is, if you also know the work by Dr. Germanot here in France, corpus callosum is an issue in fetal alcohol spectrum disorder. We expected the corpus callosum to be too thin. Interestingly, it was too thick. We're still working on the DTI data and hoping for some help here, because the processing is quite complex, but there is quite a bit of data around with us. This is the collaboration with Marseille, where they actually could show a similar phenomenon, that the connectivity within the corpus callosum in alcohol-exposed mice, babies, was abnormal. So something happens to brain wiring, something happens to the corpus callosum. It's not necessarily that the corpus callosum is too thin prenatally. It could be even too thick, but the developmental trajectory here is altered. And I think alcohol is one of the hidden epidemics, but we have many, many others. And fetal MR is really, really nice tool to look at all of those. Now, finally, this is what we might be interested as researchers, of course. How is, for instance, acetaminophen, as our US president is just claiming, impacting the generation of the brain? You could do a nice study on that, comparing groups. Of course, that's attractive as a researcher. For clinicians, we are interested in the individual. We are interested in the one lady sitting in front of us in tears, not knowing what's going to happen. And this is a difficult sort of communication situation, as you can imagine. So we are actually not interested in the basically abnormality per se. We see a spina bifida, we see a lissencephaly, but we are interested in the one exception or in the one deviation of the normal course. And one way Roxanne is trying to do that, and we have heard similar approaches throughout the entire morning today, is to generate, also synthesize templates and basically compare them to the actual case and see how those cases are actually deviating from normal. And these deviation maps might tell us then more how this individual is not fitting to maybe the the normal brain, but also a typical case of ventriculomegaly, the typical case of whatever you have. So again, to do prenatal neurology, these are tools which are extremely important to us. This is not something, you know, I'm eager to use that tomorrow. If you give it to me, I will use it tomorrow. This is an example of a case with tuberous sclerosis. You know, they have cardiac rhabdomyomas, huge tumor in the heart. This is the ultrasound. But also they have these subependymal nodules in the brain and catastrophic epilepsy. So what we did in this case is we tried everolimus, which is a medication, you know, in children works quite well with TSC cases. Seizures, it has a good impact on seizures. And we were interested in this experimental use prenatally, what happens to the lesions. And you see that the subependymal nodules were decreasing by half longitudinally. And what was very reassuring is that the cortex volume stayed within the normal range. So the cortex volume trajectory was not affected. This is an individual case, of course, but this is a hands-on practical use of what you're providing. And finally, another case, moderate ventriculomegaly. Here we are speaking of 12 to 14 millimeters, still sounds sort of large, but still most of those cases, like 80% of these children will lead a totally normal life. 80%. We are not interested in the white swans. We are interested in the black ones, which are the 20% not having these good outcomes. Now, if you do the MRI, you see that the ventricles are somewhat wide. What you also can appreciate is that there are several MR soft markers, and that's the way how we currently approach this. We are looking at, for instance, the aqueduct. It's not so easy to see that on ultrasound. Is it patent? Is it not? We are looking at the vermis. There's a vermian tail sign, as you can see here, in a totally normal positioned vermis, a very interesting finding. The corpus callosum is rather thin here, difficult to quantify again. And there is another thing, is these white spots right there. And that's a very subjective thing to us. You need to know your scanners, you need to know your images. But this is the region of, we have heard it today already, crossroads. Now, what are crossroads? These are regions where a lot of fibers are intermingling with each other. You can do field tractography and see these different types of fibers which are getting very close there together, and they are causing, for whatever reason, maybe also extracellular matrix or fluid content related, a bright signal on T2 in that area. Now, this is again a tractography, and that would be the parietal or frontal crossroads. Those crossroads could be sort of lighthouse zones. They could be indirect markers of something going on really abnormal in the brain. And in this case, it was a AB4B1 mutation, homozygous, and that is a mutation which is causing axonal transport not to work very well. So axonal transport is altered. And maybe this is going on, or surely this is going on in the entire brain, but in this C1 or wherever crossroads, they might be particularly pronounced and visual to our open eye. This is causing hereditary spastic paraplegia. And the reason why Darius Ibrahimi Fakhari from Boston Children's is laughing here, despite this kind of really serious disorder, is that they developed a gene therapy for that, which is now on trial. Basic common finding in all of that is don't start therapy like 2, 3, 4 years afterwards. You need to start it as early as possible. And I think you, me, we will see in our lifetime gene therapies applied in babies. I know this is ethically, of course, we have to discuss a lot, but I'm sure how the world is currently evolving, we will see gene therapies applied prenatally. There is just one way to do that in a way that the right therapy is coming to the right baby, is to have brilliant diagnosis. And that's why we are here. We have to kind of work on that further. So again, biological plausibility. We have to understand all these layers. It's not enough to understand genes. It's not enough to understand just the phenotype. It's not enough, I have to say, to understand MR images. You have to know all of those layers to optimize counseling, to optimize therapies, and to modernize fetal medicine. Yeah, and with that, I want to thank you for your attention. Thank you.